The topic of discussion during the symposium was drug discovery and the challenges in the path to drug discovery for anti-epileptogenic therapies. So we face a lot of challenges, largely because this is a new area for epilepsy researchers and the field. We have been very successful in developing anti-seizure medications, anticonvulsants, which are symptomatic treatment of epilepsy. However, the new direction that the field would like to go to is prevention of epilepsy and developing disease-modifying therapies. So the first step in that process is to engage the basic scientists and harness the early stage discoveries in their laboratories, which is the topic of my presentation. I'm a basic scientist, and we study how epilepsy occurs in the laboratory. We want to encourage other basic scientists to do the same and to come up with novel targets for intervention. My topic was to understand how the process occurs and to identify novel targets. I discussed a target in depth at the symposium, which is the role of redox and metabolism, which is not very well appreciated in epilepsy. What it is, it's a soluble factors that are generated by the respiration of cells and that underlie the circuit itself, and that can lead to damaging effects of neurons following any kind of injury that makes someone susceptible to epilepsy. They would not really know this very easily, and that's why we need some biomarkers so that we can detect reliably after someone has had a traumatic injury or an infection or a event that would suggest that they would be at risk for developing epilepsy. And then if we could measure molecules in their bloodstream or even in their brains by non-invasive methods like imaging, we would be able to evaluate them for their risk of developing epilepsy and maybe able to intervene so that they would not have that condition occur. So that's the overall goal of disease modification. You would educate folks in the healthcare system, and hopefully we do that well at venues like the American Epilepsy Society, where you're able to attract individuals who are working on patient populations, the general neurologists and also epilepsy specialists. The best way to do this is to get people into an epilepsy center where they would be evaluated by a neurologist who is actually an epileptologist. It is a risk factor. It is a known risk factor whether all humans who have a traumatic brain injury will develop epilepsy is not known. In fact, it's only a small percentage that actually go on to develop epilepsy. So there's many factors that play a role in contributing to the eventual disease itself, manifestation of the disease. Therefore, it's difficult to treat everybody. In terms of acquired epilepsy, it's difficult to inform patients what to avoid. Obviously, we would like to avoid injuries to the head. Head injury is a major cause. Infections are a major cause. It's difficult to prevent these sorts of things from happening. They're accidental, obviously, they're not intentional. So it's not a really, it is a public health issue, but it isn't something that can be compared to smoking or drinking, which is a voluntary issue. I would like doctors to be aware that metabolic factors play an important role in the development of chronic epilepsy. and to be aware of that and to make patients aware of these issues as well. So there may be perturbations that may not be as complex as even drug therapy, but they could be dietary methods which have been ongoing in epilepsy for a long time that could intervene at in simpler ways to modulate their disease progression. Our research is very basic research, and it is in the preclinical arena, which is the arena that is prior to a clinical trial. What we do is we identify which processes need drug intervention, and we develop therapeutic agents. We prioritize these therapeutic agents so that we can select one or several that would be efficacious in our animal studies and our cell studies, and then we try to move it forward to a clinical pathway, a translational pathway first before it goes to a clinical pathway. And so that there would be a need for toxicology studies which determine safety of the compounds, and any other studies that make them more able to, for the FDA to approve them as therapeutic agents. The hope is that the holy grail of disease modification will be realized in 10 years. So my hope is that it would be, there will be many disease modifying therapies that will be there for us and there for doctors to prescribe to patients so that they would prevent and eventually cure epilepsy. My research is pharmaceutical, which will be drug development, but there can be many ways in which this sort of therapy could be moved forward. Some of them are cell-based therapies, others are aid with the surgical methods as well, and the drug therapies are most likely going to be the major arsenal that we have against epilepsy and disease modification.